Good day, and welcome to the Batoclimab program expansion and Batoclimab update. At this time, all participants are in listen-only mode. After the speaker's presentation, there'll be a question-and-answer session. Instructions will be given at that time. Please note this call is being recorded. I would now like to turn the call over to Stephanie Lee. Please go ahead.
Good morning, and thanks for joining today's call to review Batoclimab's program expansion and an update on Batoclimab. I'm Stephanie Lee with Roivant. Presenting today, we have Matt Gline, CEO of Roivant, and Ben Zimmer, CEO of Priovant. For those dialing in via conference call, you can find the slides being presented today, as well as the press release announcing these updates on our IR website at www.investor.roivant.com. We'll also be providing the current slide numbers as we present to help you follow along. I'd like to remind you that we'll be making certain forward-looking statements during today's presentation. We strongly encourage you to review the information that we filed with the SEC for more information regarding these forward-looking statements and related risks and uncertainties. And with that, I'll turn it over to Matt.
Good morning, and thank you, everybody. Thank you, Steph. Thanks for dialing in on short notice, as always with these things. We have two agenda items today. We had originally intended to schedule a call right around now to take you through our new indication of Priovant, lichen planopilaris, and we're going to do that. And excited about that. I think that's going to be a really great addition. And then just because it happened to line up timing-wise, that we just got the phase 3 data in TED and Batoclimab, we put out a press release on that this morning, and we'll cover it for just a few minutes at the end of the call and take questions on both. So I'm going to kick it off on slide 4, starting with LPP. So look, I think it will come as no surprise to any of you that we are at this point super excited about what Batoclimab could be, and frankly proceeding with urgency around the idea that it's going to be a large opportunity to expand across multiple indications and to build, I don't love the phrase pipeline in a product generally, I think it's overused, but a pipeline in a product. We so far on slide 4, look, we've set some criteria for ourselves for the things that we really care about in terms of what indications are going to work well. You know, starting with the sort of orphan immunology space where we're looking at indications that have, call it, mid-tens to very low hundreds of thousands of patients with a lot of morbidity and sort of severe disease with a lot of unmet need. We're also looking for indications where the pathobiology aligns with our specific unique mechanism hitting both JAK1 and TYK2, and ideally where there's some proof of concept data highlighting the potential benefit of either JAK1s or TYK2s or both, and we have something specifically useful on that here. And finally, we're looking at indications where, with high unmet need, where there's really not much approved right now. So I think everyone's familiar with the sort of first 3 indications in our portfolio there. Obviously, dermatomyositis, first of them, with the PDUFA date in the third quarter, NIU with phase 3 top-line data coming in the back half of this year, and cutaneous sarcoidosis with phase 3 studies starting in the second half of this year. And I'm pleased to announce that there's another one now, lichen planopilaris. We'll talk more about the disease in a moment, where we're beginning a effectively direct-to-registrational combined phase 2b/3 program. In fact, began, I would say, last month. And so that study is now underway and represents a fourth leg to the stool here that we are really excited about. So LPP on slide 5, and then I'm going to hand it over to Ben to talk a little bit more about the indication and sort of why we're excited about it. Look, this is a severe and deeply unpleasant disease. It's a highly morbid inflammatory scalp disorder. There's nothing approved for it now, and it localizes. It's an inflammatory disease of the scalp where the inflammation localizes to the permanent part of the hair follicle, which first of all leads to generally irreversible hair loss, but is also scarring, can be permanently disfiguring, and in many cases is intensely painful, has a lot of itch, burning, redness, scaling. This is a really, really tough disease for these patients. When you see the sort of polypharmacy and some of the other things we'll talk about later in the call, these patients require a lot of medical care and have bad comorbidities. There are no FDA-approved therapies for LPP, and these patients require chronic, aggressive, multimodal therapy and are largely poorly responsive to first-line therapy like steroids and ISTs, hence the polypharmacy, hence the pain management. This is not a well-controlled disease at all. And it's a fairly prevalent disease. It's got sort of a large orphan size. It's I'd call it DM-like in population size with probably up to 100,000 U.S. patients, and the literature indicating the prevalence and diagnosis is increasing over time. So we think this is just right in the center of the bullseye for us. It's the kind of disease, you'll hear more about our understanding for why our mechanism is good for it, but the kind of disease with high unmet need and a lot of severity that we think Batoclimab is exactly the right kind of drug for. So with that, I'm going to hand it over to Ben, starting on slide 6, just to talk through what the experience of these patients is like and a little bit more about why we're excited about it. Ben, take it away.
Great. Thanks, Matt. So as Matt mentioned, an indication with very high patient burden. A lot of that is the direct symptoms and manifestations of the disease, as Matt walked through. Also on slide 6, you can see LPP is associated with an increased risk of many severe comorbidities, including both skin cancer and other autoimmune diseases. And, as Matt mentioned, no FDA-approved therapies. Given the severity, a lot of different drugs are attempted off-label, but generally with limited efficacy and high rates of discontinuation based on tolerability and efficacy issues. So really an area of very high unmet need, very high sense of urgency to treat quickly and with efficacious therapies, and we think there's really a significant opportunity for a new efficacious drug. And, turning to slide 7, we're optimistic that brepocitinib can be that drug. Like cutaneous sarcoidosis, LPP is a disease driven primarily by Th1 polarized T cell aberrant behavior, and this is a category of indication where the biology really aligns with TYK2 JAK1 inhibition, interferon gamma, and IL-12 are two of the critical cytokine signaling cytokines within the Th1 pathway, and a JAK1 TYK2 inhibitor is distinctively able to suppress signaling of both of those cytokines. And then, I bring up cutaneous sarcoidosis because through that study and other phase 2 studies, we've really seen that play out in the clinic with excellent data from brepocitinib in indications with similar biology. And so mechanistically really feel this fits into the sweet spot of where brepo can be a potentially highly efficacious treatment. In addition to brepo specifically, there's a large number of case reports and investigator-initiated trials of both JAK1 and TYK2 inhibitors that generally establish clinical validation for this mechanism and gives us the excitement around moving quickly and rapidly into a potentially pivotal program. In turning to slide 8, one of these is actually in brepocitinib itself. This was a small investigator-initiated trial conducted at Mount Sinai. Unlike many of the other investigator-initiated trials, this one was placebo-controlled, though with only 3 patients in the placebo arm. It was also using the LPP-AI, which is a generally noisy instrument that is actually not that preferred by clinicians, and so in our program we're using different endpoints. So small study, really not to be overread into in terms of the specific data, but big picture, you see the brepo treatment arm clearly getting better over time, and really overall, amidst the totality of other data, furthers our confidence around the POC for this drug and moving quickly into a potentially pivotal program. And before Matt walks through the design of our trial, I would also just note on slide 9 that although the clinical data with the small study and the LPPAI is I think around a somewhat noisy endpoint, though still persuasive. Actually, what I think is probably most powerful about this study result is the biomarker data, which is on slide 9, where you see very clear and convincing effect of brepocitinib on multiple markers of Th1-driven disease activity, including both interferon gamma and IL-12 themselves, as well as other important chemokines that are markers of Th1-driven inflammation, like CCL5. So, I think all of this together sets the foundation for what we view as a high probability of success study in an indication with very high unmet need, and we're really excited to be moving very quickly into a potentially pivotal program that should hopefully deliver some excellent data. So with that, I'll hand it back to Matt.
Thanks, Ben. Appreciate it. So on slide 10, you can see the trial design that we've initiated for brepo and LPP. And this is really designed to function like a single straight to registrational trial, except, and this is sort of there are many benefits to pioneering new clinical development areas. One cost to pioneering new clinical development areas is there's some work you generally have to do from a regulatory perspective. And so this is set up effectively as a sort of combined phase 2b/3, where we have a 72-patient phase 2b portion of the study. And then immediately at the end of that, we will just go straight into enrolling patients in effectively the same study into a phase 3 pivotal part 2, where we expect the end to be approximately 270, although we'll do a sample size re-estimation after the phase 2b. And that will allow us to go through sort of proper endpoint validation and to get the data we need once we ultimately do read out the phase 2b portion to finalize the regulatory work and read out the phase 3. So this should be, we're not ready to guide today on enrollment. Obviously, we've gotten a lot of enthusiasm from the investigator and patient communities as we've gotten the program up and running. Ben and the team have been engaging super actively with this population, with this population of physicians. We're not ready to guide today on timeline, but the plan here is for this to function like a straight to registrational program, and we're excited for what that means for timelines and what that means for our opportunity to get a new option to patients quickly. So we're looking forward to playing that through. Look, on slide 11, just to summarize, and I think we've hit these points all pretty clearly here. First of all, this is a disease with tremendous high unmet need. I'm confident as the investor community does work on it, talks to physicians, talk to patients, you'll see a disease with a high burden and a lot of unmet need. These patients are very uncomfortable. We have a drug that is distinctly suited mechanistically to LPP. LPP has a Th1 dominant immunophenotype. It's really sort of exactly down the fairway of where we've succeeded elsewhere. We think we've got a creative, aggressive development strategy that will get us to market hopefully quite quickly. And we think we've got good synergy from a sort of commercial perspective in the sense, not just in the sort of literal cost perspective, meaning, but that we know these docs, we know these centers, we know this community. Priovant has done a phenomenal job, Ben and the team, of building relationships, at tertiary medical derm centers in some of these other indications, and we think we're really going to be able to leverage those relationships. In fact, they were some of the very same relationships that help us sort of key in on LPP as a desired indication to begin with. So really looking forward to that. Super appreciative of all the work the Priovant team has done, and excited to announce that trial is now up and running. And so looking forward to sharing more updates and answering questions about it today and in the future. Before we go to Q&A, I just want to turn quickly to the next topic, which is the phase 3 study results in TED. So as I'm sure many of you saw, Immunovant announced this morning, it's on slide 13, that our phase 3 TED studies had failed to meet the primary endpoint for Batoclimab. As a reminder, I think everyone's clear on this, Batoclimab is the first generation anti-FcRn antibody at Immunovant. The subject of the vast majority of our future, all of our future development at this point is IMVT-1402. So this was effectively the last study to read out from the first generation program. It was not a focus area for us for that reason, as we've said in other contexts. Obviously disappointing to see the study fail to meet the primary endpoint. Batoclimab had succeeded, as you may remember, in an earlier phase 2 study in TED. Ultimately, in the long arc of our future, I suspect the meaning of TED for us is largely going to be that it was what guided us to Graves' disease and got that program up and running. And we'll talk more about some of the data we generated there. But first, just really quickly on what we saw in the study. So as a reminder, this study was actually designed pretty similarly to our phase 2 study in Graves. It had a 12-week period of high-dose Batoclimab designed to get IgG as low as any FcRn can take it, followed by a 12-week period of lower dose Batoclimab, which suppresses IgG in general, more like what we see in some of the other FcRns currently on the market. And we actually have that number later in this deck in terms of what we saw in that period, at least in a subset of patients. The primary endpoint of the study was 2 millimeter, greater than or equal to 2 millimeter proptosis responder rate, and that's the endpoint that we failed to meet. We measured some secondaries as well. What we said in the press release, and I just want to reiterate here again, not trying to dress up a failed study. This is not a study that supports sort of further progress in TED. But in terms of things that we were looking for, there were probably 2 key things we were looking for in this study other than success. One was continued evidence that our deeper IgG suppression matters. And I think there's like a few ways to read that in this study, but I'll say first of all, consistently across everything we looked at in the study, patients did better in the first 12-week period broadly than in the second 12-week period. again, it was a relatively, noisy study in terms of some of the endpoints, but there's a lot of different data points pointing in that direction. We take a lot of signal from that, including, frankly, the fact that at least one other FcRn inhibitor failed on futility in TED, and we would not have failed on futility. We saw separation in a positive direction in multiple endpoints, and we'll talk more about that in a second. And then the other thing we were looking for is evidence of read-through to Graves' disease and trying to understand, as this, although it was not a study focused on hyperthyroidism, in fact, very hyperthyroid patients were not allowed into the study. There were nonetheless a relatively small proportion, but large in the context of studied hyperthyroid patients with FcRns of hyperthyroid patients, and we wanted to continue to confirm our hypotheses there, and I think we've had some good outcomes there too. On slide 14, there were a lot of different data points we could have shown in terms of like the benefit in the study, and I think anyone who's familiar with TED will look at these numbers and agree. Numerically, in the context of treating TED patients, these are not like super exciting proptosis improvements, but we did see, meaningful numerical separation from placebo on, for example, change in proptosis at week 12. And in fact, when you pulled the two studies together, that was sort of nominally significant in a post hoc statistical analysis. Again, with that and $20 you can buy a sandwich, but I think it is evidence that the drug was, in our view, doing something and that we were seeing a benefit for these patients. Probably most importantly, these numbers all got worse, and I don't have that slide in here, but these numbers all got worse as you went from week 12 to week 24. So as you step down FcRn dose and saw lower IgG, the amount of proptosis improvement degraded, which I think is an important point. I think also, as we think about this patient population, this was an active TED patient population that has sort of later stage, more advanced TED, the kind of populations that have been studied in other TED programs, by the way, many of which have, I'd say, underperformed recently, as I think the disease landscape of TED has changed a little bit. But there is plenty of evidence in this data set, including the slides you're looking at on slide 14, that suggests that we will improve proptosis and potentially delay proptosis development in Graves' patients. And I think that is important to us as we continue to measure all of those things in the ongoing Graves' disease program for 1402. And I think the study does broadly support that we should be able to deliver a benefit of that kind in our Graves' program. And then the last thing, which is probably the single most important thing to come out of this data set in terms of read-through to Immunovant's prospects from here, is on slide 15, which this is, on the left-hand side, the pooled results across the two studies, and they're pooled simply because the ends are small overall, of the hyperthyroid patients in the program. Again, the study didn't allow very hyperthyroid patients, but within the boundaries of what was permitted, there were about 20 hyperthyroid patients between the two active treatment arms of the studies. And what you see here is, in those two studies as pooled, there was a 75% mean IgG reduction. So Batoclimab did consistently what it has done in all of these studies from an IgG suppression perspective. And actually what we saw was an 80% responder rate using the same responder definition, normal T3 or T4, at least T3 and T4 below the upper limit of normal, with no increase in ATD doses. This, again, the same definition we used in the phase 2. And we said 80% responder rate, which was just bang on the same at the end of week 12 as the responder rate we saw in the more severe Graves population in the phase 2 study for Batoclimab. And one thing that's comforting is this was a very different patient population, a very different study design. Remember, a very different study sort of structure. The phase 2 was a single-site study with no placebo, whereas this was a placebo-controlled study with many patients across many different centers and a different hyperthyroid population. So to see that consistent effect, especially at the high dose, is helpful. I also think it's notable, as with the phase 2 study, we saw a lower responder rate after the 24-week period, after the second 12-week period, as we reduced IgG suppression. And in fact, nicely, although I can't claim given the end there's like a perfect correlation, the IgG suppression wound up a little bit lower in that population, and the responder rate wound up a little bit correspondingly lower in that population. And I think that just further underscores that in a disease like Graves' disease, the deeper you can get IgG lower, the better you're going to do with these patients. So I think that is also, again, a helpful outcome from an otherwise disappointing study. So look, again, there was a lot of debate here about how much to say about this given the outcome, which is disappointing for these patients. I want to say, as I'll say again at the end of this, thank you to the investigators, to the patients, to the Immunovant team. These studies are hard to run whether they work or not, and everyone trusts us with their care. And I'll say, I think the useful scientific evidence that came out of this on, supporting our theses around treating proptosis via Graves, and on deeper IgG suppression mattering and being able to treat Graves patients will certainly help inform our plans. Notably, we're only sharing a relatively small amount of this data now. A thing that we've said consistently is that we think there is important and useful information in this study that informs how we are managing the Graves study super actively. And so I expect we will share more of this over time, but for now we're being relatively quiet in terms of the breadth of what we're sharing. Happy to take questions broadly though. Look, to wrap up for the day before we open it up to Q&A on slide 16, our pipeline to me looks better and more mature every time we get on the phone. I'm really excited about the addition of LPP here. Brepo is really shaping up to be a broad franchise opportunity across multiple indications, and I think there are potentially many more lines to add to this graph as we think about where we're working right now, and the Priovant team has done an awesome job of executing thus far on clinical programs and excited to see the outcomes from this study and more. I'm really happy with where the FcRn franchise looks. Obviously, IMVT-1402 firmly the drug to beat there, and excited about the data coming later this year, both in D2T-RA and in CLE, and excited for 2027, which is, one of the, if not the most important years, at least in Immunovant's history, and potentially in Roivant's, between the Brepo launch and the Graves' disease data that's coming. Not to mention, which won't be subject to this call, mostly Sigrowat with the phase 2b data and PH-ILD coming the second half of this year. So a rich catalyst calendar on slide 17, a bunch of stuff upcoming that we're excited to talk about. Just a huge year for us, and disappointed about the outcome on TED, but otherwise just really excited about what we're doing here. So I'm going to stop there. I'm going to open it up to Q&A. Excited to take questions on LPP. Obviously, also happy to take some questions on the TED study as well, as I'm sure people have them. And so with that, I'll turn it over to the operator for Q&A.
Thank you. If you'd like to ask a question, please press star 11. If your question has been answered and you'd like to remove yourself from the queue, please press star 11 again. Our first question comes from David Risinger with Leerink Partners. Your line is open.
Thanks very much, and thanks for the updates, Matt. So I have two questions. First, for LPP, slide 8, I don't know if you have these details, but could you talk about background therapy in the 13 Brepo patients and the 3 placebo arm patients, and whether that might have impacted the results of that study that was shown on slide 8? And then I know that Graves' disease is actually a comorbidity in a meaningful percentage of LPP patients, which obviously is a positive indicator for potential success of your new program, given the Graves' disease results that you've previously disclosed. But could you talk about that and any other comorbidities that we should be aware of with respect to LPP? And then, actually, I have one more after that, if that's okay.
Thanks, Dave. Appreciate the questions. So on the first one, for the technical answer to that question is that meds were washed out pre-baseline in that IIT. That said, in general, I want to highlight LPP patients are polypharmacy patients who are on a whole host of background meds, and one of the things the Priovant team has thought very carefully about is management of, call it, polypharmacy background and concomitant meds in the phase 2b/3 program, simply because these are very sick patients and poorly managed. There are a ton of comorbidities with LPP. These patients are sick in a variety of ways, including with Graves'. As a reminder, the Graves' study was obviously on the FcRn side, and this study is with brepocitinib, JAK1, TYK2. So I think the ability to effectively access and treat Graves' patients is probably a useful indicator of a sick patient population, but obviously in terms of the actual pathobiology, probably not as much direct read-through. But nonetheless, look, I think excited about what we're going to be able to do for these patients. And frankly, look, I think Brepo is a great drug for patients with a lot of comorbidities because of the broad anti-inflammatory activity.
Great.
Dave, you said you had one more.
Yes, thank you. So obviously disclosed that the subset of hyperthyroid patients in the TED study showed similar response rates of thyroid hormone normalization to those seen in the Batoclimab phase 2 in Graves'. So how did Batoclimab perform in thyroid eye disease in that subgroup relative to the rest of the study participants?
Yeah. I think the answer to that question is somewhat better in the hyperthyroid population in general. And we saw the most recent cut of that I looked at in detail was in only one of the studies, so I don't have the full pooled data on the top of my head. But in general, the answer is we did better in hyperthyroid patients than in the general population on proptosis. Thanks, Dave.
Got it. Thank you very much.
Thank you. Our next question comes from Yasmeen Rahimi with Piper Sandler. Your line is open.
Hi, this is Liam on for Yaz. Thanks for taking our question. So I guess just in regard to the TED study, do you think there's an opportunity to transition any of the TED sites to the ongoing Graves pivotal program? And then if you could just provide an update on how enrollment is progressing across those two trials as well.
Yeah. So first of all, specifically in the Graves program, we're focused on Graves patients, and we've excluded the more moderate and severe TED patients. And many of the sites in the TED study were more opto-focused sites and sort of TED-focused sites, whereas obviously we're focused on endos and the sort of Graves treating physicians. That said, I'll say also we are not particularly thirsty for more sites in Graves right now, just because enrollment is generally going well. We're reaching as many docs as we can, and we will certainly increase site numbers over time in the studies. But in general, I'd say we're happy with how enrollment is going in Graves and seeing a really enthusiastic reaction from the doc community there. Again, we expect both Graves studies to read out next year. Thanks, Liam.
Thank you.
Thank you. Our next question comes from Sam Slutsky with LifeSci Capital. Your line is open.
Hi, this is Kate on for Sam. Thanks for taking our questions and for the update this morning. Yeah, we were curious just regarding the TED study, how much of a difference in thyroid response was there in the first 12 weeks versus the final 12 weeks? And I guess, did you see any similar numerically better trends on the secondary endpoints? And I also have a follow-up.
Yeah, great. So I'm sorry, I didn't quite follow the first question. I think it was the difference between first week and 12 weeks and second 12 weeks. I didn't quite catch if you asked on the primary or on the endpoint that we showed in the deck. The answer in either case is there was degradation in both numbers as between the first 12 weeks and the second 12 weeks. The proptosis responder rates, I think in one of in the blend across two studies, the proptosis responder rates in the treatment arm were like low to mid 20s, maybe low 20s%, and the placebo arm was sort of high teens. And so, there was some degradation, but the numbers of responders were relatively small. In general, we saw trends supporting better performance in the first 12 and the second 12. I think the proptosis the proptosis improvement measure that we showed was something like, I'm trying to do math at the top of my head, which is always dangerous, but like 20 to 30% better at the end of week 12 than at the end of week 24. But we get the exact number with a better thing. And the answer is yeah, it was consistent across both the primary and multiple of the secondary endpoints that people performed better at week 12 than at week 24. Thank you.
Okay, great. And if you don't mind if I squeeze one more in, for the subset of hyperthyroid patients, any anecdotes of how they're doing off treatment and whether they're remaining euthyroid?
It is a great question. I don't have any anecdotes from that population right now, but it's a good question, and honestly, I hadn't thought to ask in the last couple of days, but we'll take a look.
Thanks so much.
Thank you. Our next question comes from Brian Cheng with JP Morgan. Your line is open.
Hey, Matt. Thanks for taking our questions this morning. Just catching up on your comments around LPPAI being noisy. How should we think about the correlation of the endpoints that you have mentioned on the slide here, and also the primary endpoint that you'll be using for the phase 2b/3? And then I have a follow-up. Thank you.
Thanks. It's a great question. IGA 0/1 is generally just a much higher bar than LPPAI. But Ben, do you want to talk a little bit about endpoints and where we're at?
Yeah. The LPPAI, it's a composite endpoint that includes a bunch of different symptoms. Some are physician assessed, like erythema and scale, and some are patient reported. The biggest problem with the endpoint, in addition to just combining all of those, like all composite endpoints can have noise, is that there's no definitions in the LPPAI. Each of the symptoms is rated 0 to 3 with no guidance or definition for the raters on what would be a 0, 1, 2, or 3. So generally just like a highly noisy measurement. What we've done instead is really try to break it up into much more precise measurements. So our IGA measures erythema and scale with clear definitions consistent with the rigor that goes into an FDA supported IGA, and then through different secondary endpoints we'll be assessing each of the different other burdensome symptoms for patients like pain, itch, etc. Through generally speaking NRS scales. So I think in our view a more kind of less noisy and more clinically meaningful to physicians and patients way to assess benefit.
Great. And maybe just follow up on the TED data they presented today. Can you also give us some color on the proptosis improvement that you saw once you stepped down to the 340 mg/s portion? Particularly, was the proptosis impact quick to see deterioration once you pull back on the dose? And how does that proptosis reduction change once the IgG also start to see lowering once you switch to the step down dosage?
Yeah, thanks. Look, I think the answer, and I don't have the number in front of me. I've seen it in the last couple of days, but the answer is the proptosis improvement that we showed in the deck gets worse at week 24, and I think it's about 20 to 30% worse at the end of week 24 relative to at the end of week 12. So that's about what it looked like specifically on the improvement. So that's and I don't have a time scale of like how quickly it degraded, but in general IgG falls over a period of a couple of weeks to the lower level, and so I think and then proptosis obviously lags further. So I think that it takes some time to get worse, roughly. Yeah. Thanks, Brian.
Okay. Thanks. Thank you, Matt.
Thank you. Our next question comes from William Pickering with Bernstein. Your line is open.
Hi, thanks so much for taking my question. So on LPP, can I just confirm that the primary endpoint for the phase 3 portion is IGA 0/1 responder rate, and kind of what are your expectations for effect size there? And then could you talk about how you're going to be handling background therapies in the study? Thanks.
Yeah, thanks. Great question. First of all, I'll just say, and I'll hand it over then to Pine too. I think, first of all, the primary of the phase 2b portion is IGA 0/1. I think part of the reason the study is designed this way is to make sure that we're aligned with the FDA on the phase 3 primary. And so, I think our hope and expectation is that there'll be identical endpoints in the phase 3, but obviously until we've finished the phase 2b and had a conversation with FDA, we probably don't know 100% for sure. And then in terms of background meds as well, it's just to confirm my understanding there and anything else. Ben, do you want to answer those questions?
The general approach is going to be to wash patients out of background meds ahead of the enrollment quite aggressively, which is consistent with how we've approached a number of other trials like cutaneous sarcoidosis, and things should set it up for success. And yeah, I think our base case is that the primary endpoint in the phase 3 will be the IGA 0/1 with 2-point reduction. That's obviously generally the gold standard derm endpoint for inflammatory conditions. But as Matt noted, part of the point of this is an endpoint that's never been actually used before. So part of the value of the phase 2 portion is to get a sense of the behavior of the endpoint, including even just on a blinded pooled basis before we read out the phase 2b, just to confirm that thinking ahead of starting the phase 3.
Thanks. And if I could just squeeze in a follow-up, what do we know about the efficacy of off-label JAK use in this indication?
Yeah. I mean, look, I think among the things we know about the efficacy of the use of JAKs and drugs like ours, obviously, is what we showed for the IIT for the use of brepocitinib. There's a lot of case reports as well. I think in general, you'd see across the board, like broad support for mechanisms like these, JAK inhibitors, TYK2s, and certainly JAK1s and TYK2s combined in improving these patients. I think, they're not like, in my sense, not like super widely used off-label, but they seem to work based on case reports.
Thank you.
Thank you. Our next question comes from Samantha Semenko with Citi. Your line is open.
Hi, good morning. Thanks very much for taking the question. Two for me. Just another on the IGA 0/1 endpoint. I think, Ben, you mentioned that this will be the first time you're using this endpoint in LPP. Can you talk a little bit about some of the powering assumptions in the phase 2b and what kind of placebo response you're sort of expecting to see or designed maybe around to see? And then, Matt, you outlined in the start of the call some criteria for assessing indications for brepo. Just wondering what your capacity is for nominating even more indications going forward. And is it reasonable to think that you might stick within the rheum derm sort of inflammatory space for those additional indications? Thanks very much.
I'll take the second question because I can answer on Ben's behalf and put pressure on Ben, which is, look, I think we are almost endlessly enthusiastic for the breadth of opportunity for brepo. I think there's a lot of value to the relationships we've built in the rheum derm context, and frankly, a long list of indications in the rheum derm context that we like. So I think that you will see us do more there. I also think we are excited about indications that go beyond that context, and you may very well see us go kind of outside of that area as well. I think we want the Priovant team to succeed at everything it's doing, but certainly, this will be the second additional pivotal study we're starting this year in addition to the cutaneous sarcoid one. So, we're looking at now, assuming everything succeeds, at least 4 launches over the next couple of years, and I hope we can add to that list. Ben, you want to take the question on IGA 0/1 powering?
Yeah. So as a general matter, we've looked at the kind of precedents we've used. If you look at kind of how these instruments work in general, the IGAs, particularly those that are developed in partnership with the FDA and used for registrational programs, the placebo rate in general across them tends to be extremely low. We saw that in our own phase 2 sarcoid study recently, particularly requiring not just a 2-point reduction, but 2-point reduction to 0/1. That tends to be a very high bar for placebo. So, our general ingoing assumption is that we would hypothesize the placebo rate would not be high, but we'll have to see what the actual results are. We think that on the basis of looking at effective therapies in other inflammatory skin disorders and kind of how the IGAs there have behaved, that this phase 2b portion should be very well powered to detect a difference. But again, ultimately, to Matt's point in the opening, from a clinical confidence perspective, I think we would have been happy to just go straight into a phase 3 program without doing a phase 2 piece. But I think that just given we're pioneering a new indication here, part of the point of the phase 2 program is really to learn more and inform the phase 3, and ultimately we'll see the results of the phase 2 and use that to repower the phase 3 if needed, which is what will ultimately be the portion of the study that we rely on to support potential registration.
Great. Thanks very much.
Thank you. Our next question comes from Yaron Werber with TD Cowen. Your line is open.
Hi, guys. This is Sarah on for Yaron. Thanks so much for the color in this presentation. Just 2 quick questions from us. So On the brepo study, which of the 7 other successful phase 2 studies give you conviction in LPP, mainly on the beyond IGA 0/1, which I know you've just discussed a bunch, and maybe on the secondary endpoints, and then just a follow-up beyond that?
Yeah, perfect. Look, obviously, alopecia and CS, which we highlighted in this deck, are both sort of very much the same phenotype from an inflammatory perspective. They're both sort of really sort of Th1-driven diseases. I'll say, personally, I also just take a lot of comfort from the overall breadth of clinical evidence at this point in inflammatory disease across, you look at the interferon drivers of LPP, et cetera. Like, I just think, like, it's pretty clear, and you can see it in the IIT on the sort of biomarker data. It's pretty clear that we sort of hit a lot of the right biology here, and I think that gives us a ton of comfort. And then, there's many of the other indications, significant components of these same inflammatory drivers. And so I think almost all of our studies contribute, probably the ones with skin components most of all, but not uniquely. But alopecia and CS are probably the 2 most important ones.
Got it. That makes a lot of sense. Thank you. And then on the TED study, are you also taking forward the 680 and 340 mg dosing in the Graves study? And maybe just if you could just provide a little bit more color on what read-through that might have on the trial design for the Graves study. Thank you.
Okay, got it. And as a reminder, so the equivalent doses of 14, so the TED study that we read out here was in Batoclimab, which is not the drug we're studying in Graves' disease. We're studying 1402 in Graves. The equivalent doses in Graves are 300 and 600 versus 340 and 680. Our Graves programs have both 300 and 600 milligram dosing arms as between the 2 studies, and we're carrying forward both of those. As a reminder, based on our phase 1 work, our expectation would be that 300 would have, competitor-like suppression of IgG, and 600 would have IgG suppression that is similar to what we saw at 680 milligrams in the Batoclimab study. And I think, all of the data we have is small n, et cetera. But the short answer is that our best expectation is that the phase 3 data is heavily informed by what we saw in the phase 2, which is to say the high-dose arm outperforms the low-dose arm, and that we see sort of adjusted for differences in endpoints and populations and other things, similar results at the high-dose arm in the phase 3 to what we saw here. As a specific reminder, the longer 52-week 2502 study is 600 milligrams only, and then the shorter 2503 study, the 24-week study, is 600 versus 300 versus placebo. So that's the difference. Thanks for the question.
Thank you so much. Yeah.
Thank you. Our next question comes from Thomas Smith with Leerink Partners. Your line is open.
Hey, good morning. Thanks for the updates and for taking our questions. First, on the TED results, could you comment on what you observed on the TRAb levels in this study over time and how those compared to the Graves' disease data set? And then can you clarify, were the hyperthyroid patients in this study permitted to titrate their ATD dose? And if so, did you have any patients that were able to down titrate or discontinue their ATDs entirely, either over the course of the 12 weeks or the 24 weeks? And then I have a quick follow-up if I could.
Yes. So on the TRAb levels question, I guess, like, the first thing I'd say is, like, the data was just, like, unsurprising and matched what you would expect from these studies. So I'd say, yeah, that's probably the best thing to say about TRAbs. On ATDs, the answer to your question is no. In fact, they were not permitted to titrate ATDs. So these patients were required to stay on a stable ATD dose for the duration of the TED study. So unfortunately, we don't have evidence of clinical practice around ATD titration here. In fact, not only they effectively weren't permitted to titrate ATD. So we don't even get a look at what they would have done organically with these patients. Now, as a reminder, and I think in some ways the following is comforting and helpful. These were different hyperthyroid patients than in the phase 2 Graves study. In the criteria required patients to be relatively close to euthyroid. And so these patients were less sick hyperthyroid patients than the hyperthyroid patients in the phase 2. I think the fact that they continue to respond at a similar rate is encouraging insofar as it highlights our ability to treat a pretty broad range of Graves patients at this point.
Got it. That makes sense. And then just one quick follow-up, if I could. On looking forward to the DQ-TRA results later this year. Just wondering if you could provide any updated expectations for that readout, and also any clarity on whether you expect to report both the Part A open label and the Part B randomized withdrawal portion simultaneously, if you're thinking that'll be more of a staggered approach. Thanks so much.
I don't have updated guidance to give on either of those questions right now. We're still working through our analysis of the sort of best criteria to run the phase 3 and so on. I'd say that work is happening actively, and we'll share our thoughts on it as soon as we're ready. But we're excited about it too. Thanks, Tom.
Thank you. Our next question comes from Dennis Ding with Jefferies. Your line is open.
Hi, good morning. This is Anthea on for Dennis. Thank you for taking our questions. We have two. First, the investigator study looked at other scarring alopecias like FFA and CCCA. It seems like there were promising signals there, but curious what your interpretation of that data is and why you chose to go into LPP specifically. And then second, on LPP specifically, how does disease activity fluctuate over time? And how are you planning to capture brepo's efficacy within 24 weeks, and if there are any sorts of enrichment that you plan to do? Thank you.
Yeah, thanks. I appreciate both questions. On the first one, in terms of breadth of indication, first of all, both across other lichen planus-related diseases as well as other potential scarring alopecias, there's lots of interesting data available across different studies, et cetera, that suggest opportunity, including in the IIT, as you mentioned. LPP was pretty clearly to us like the initial greatest unmet need, and a well-circumscribed orphan population that, among other things, the regulators were excited about as well. So it just felt like a nice, clean opportunity all around. But if your question is, are there even more indications in which brepo might work based on the IIT and otherwise, you're never going to get an objection to that question from me. Over time, there's a lot of different things we could study. LPP tends to have a fairly steady, unremitting course. These patients, look, the disease is ultimately irreversible in a lot of the ways it affects people, and so these patients just get worse. And so there's a high desire to treat quickly, which is something that's sort of interesting about the disease, again, because it's sort of it's effectively viewed as an emergency. It's an urgent condition. And it tends to be irreversible.
Got it. Thank you.
Yeah, thank you.
Thank you. Our next question comes from Corinne Johnson with Goldman Sachs. Your line is open.
Good morning. This is Eric on for Corinne Johnson. We have one question regarding TED. So how should we think about the implications this clinical failure has on how physicians and payers think about utilizing 1402 in Graves, given the overlap in this indication and that TED is downstream of Graves' disease?
Yeah, it's a good question. Look, I think the easy answer for me to give is overall, I think there's going to be a lot of excitement for Graves, and I think the data that this study showed on improvement in proptosis, as well as the data that this study suggests to me we will show on the same in the Graves study, I think will ultimately be encouraging for the use of these drugs in Graves. That is, I think, the limitations of using FcRns in TED mostly stem from catching the patients too late at a time where FcRn therapy is simply not sufficient to treat at that point, presentation of the disease. But I think there's plenty of evidence in this data to suggest we're going to be able to benefit proptosis in Graves patients and catch it early. Look, obviously, at some level, if this study had been extraordinary, I would be here telling you how great the read-through was to doc enthusiasm for Graves. And so, I think it probably would have been even better if this study had shown better proptosis response rates. But I think in terms of our ability to improve proptosis in Graves patients, overall, I would call the evidence from this study net encouraging. And I think in the fullness of time, docs will see it as such.
Thank you.
Thank you. Our next question comes from Delma Coyote with Guggenheim. Your line is open.
Hi, thank you for taking our question. So on ATP, you describe it as a strategic fit with the dermatomyositis, given the overlapping prescriber basis. Can you quantify the extent of that overlap between rheumatologists and dermatologists treating DM versus those treating LPP? And how does that inform your commercial infrastructure build out? Thank you.
Yeah, great question. So look, I think the short answer is LPP is mostly treated by derms, and especially hair loss and scalp experts. But there's a lot of overlap at the centers. So many of the centers that are sort of big myositis treatment centers are also big centers treating LPP. I'll tell you, and Ben should feel free to add anything to this than Gaz. Our focus on commercial build out right now is succeeding in dermatomyositis and making sure that we have enough breadth and enough coverage, enough relationships to succeed there. I don't think we are changing anything about our DM commercial strategy in anticipation of CS or LPP or anything else. I think in the fullness of time, the relationships that we have at these tertiary centers will definitely be helpful with the subsequent build outs for the other indications treated at overlapping centers. But we're going to take each of these launches as the opportunity that it is and make sure we invest fully in it. Ben, anything you'd add there?
Yeah, I'll just add on DM specifically, there's a combination of rheums, derms, and neuromuscular specialists who all will be potential prescribers, and we're focused on all three of those groups as appropriate. As Matt mentioned, I think there's overlap. Obviously, the clearest overlap is in the derm subset of that DM, that DM prescriber base, but also even a lot of the rheums and neuromuscular physicians, not all of them, but many are at tertiary centers of excellence where there's often, multidisciplinary myositis clinics and myositis specialists, especially in the rheum derm space, but also involving neuromuscular experts. And so I think there's, in addition to specific prescriber overlap, overlap in terms of overall institutional engagement and collaboration that is important.
Got it. Thank you. That's very helpful. And maybe, if I may, another question on what's your on LPP? What's the internal bar for success in phase 2B to progress to phase 3?
Yeah. So the short answer to that question is we are going to enroll patients in the phase 3 before we read out the phase 2B. So we're really not viewing this as run the phase 2B, get the answer, run the phase 3. Mm-hmm. We're really viewing this as like a continuous study. And I think the reason for that is, as Ben said, clinically, I think we have plenty of confidence to go directly into a registrational program. We're just making sure we have all the information we need from a regulatory and process perspective, as well as to get the right powering assumptions and so on for the phase 3 portion of the study.
Got it. Thank you.
Thank you. Our next question comes from Alex Thompson with Stifel. Your line is open.
Hey, great. Appreciate the update and thanks for taking our questions. I guess for Batoclimab, I think, Matt, you alluded to maybe sharing some more data from this study in the future. I was curious if you could elaborate onto whether we should expect to see more data from MG or CIDP in particular in the future. And then as a follow-up to that, curious about your thinking on whether MG and CIDP data or even some of this TED data could help support ultimate 1402 filings in the future. Thank you.
Look, I think never say never in terms of publishing MG or CIDP or other data. And, we have an interesting treasure trove of things there. That said, like the 1402 MG program, for example, reads out next year. And I think at that point, everyone's going to be much more interested in the ins and outs of that data set than anything about what we saw in the Batoclimab study. CIDP, there's a little bit more time between now and when the studies come out, and there might be interesting things to say there. So, it's certainly possible. We're obviously principally focused on 1402. I don't think we will need any of the MG or the CIDP data to support the regulatory filings. I think the 1402 data will be sort of sufficient in and of itself. Obviously, the FDA is aware of all these studies, has seen safety data and everything else from these studies. And although I'm not sure the FDA would publicly declare that they take great comfort in sort of cross-mechanism comparisons of things. Obviously, the body of evidence suggesting safety and efficacy for FcRns in those diseases is helpful to us in our interactions with the agency, and I suspect helpful to the agency in making approval decisions. Obviously, the CIEP trial design is different for 1402 than the phase 2b was, so that's something to take into consideration. Thank you.
Appreciate it. Thank you.
Thank you. Our next question comes from Douglas Tsao with H.C. Wainwright. Your line is open.
Hi, good morning. Sorry. Thanks for taking the questions. I'm just trying to understand the sequencing on the LPP phase 3 study. It sounds like this is going to be sort of continuous enrollment from phase 2 to phase 3. I guess I'm just trying to understand at what point you are going to validate the data and engage with the agency just in terms of, finalization on the primary endpoint.
Yeah, after we've read out the phase 2b, when we have the data to share with them is the answer to that question. Probably along the way as well, but after we've read out the phase 2b.
Okay. And so there will be patients who I guess you'll just be insured or just in terms of making sure you're collecting everything that is necessarily needed to input into some kind of composite endpoint if some kind of change or tweak is needed?
Yeah, the short answer to that question is yes. I don't think we are anticipating like major changes to the way the endpoints are structured and things like that. Obviously, there's a fair amount of knowledge about this going in. It's mostly about sort of validating assumptions that we're going in with.
Okay, great. Thank you.
Thanks.
Thank you. Our next question comes from Dina Ramadane with Bank of America. Your line is open.
Good morning. Thank you guys for taking our question and for the update this morning. First is maybe to clarify an earlier point. Given there is considerable overlap between the TED and Graves populations, and it looks like you guys had a strong responder signal in TED patients with elevated thyroid levels, how do you plan to apply these learnings to your pivotal Graves program? Just curious what your thoughts are on how you're thinking about the inclusion of patients with ocular symptoms, and if you think there's still an opportunity in the Graves program to show a benefit on proptosis and maybe a subgroup population. And then just on the subgroup data you presented in TED patients that were hyperthyroid at baseline, could you provide just some additional color on how this patient population compares to the phase 2 Graves in terms of kind of disease characteristics that define severity, maybe baseline, T3, T4 levels, or length of time uncontrolled or hyperthyroid on ATD? Thank you.
Yeah, perfect. These are all great questions. Thank you. Look, I think the first answer is, although this comment may read as glib, the quality of the response in hyperthyroid patients in the TED study mostly just validates our view of where we're at in Graves. That is, this is across multiple sites, a broader population in some ways, like continuing to show just like very strong performance in treating hyperthyroidism in these patients. The TED study was designed to exclude severely hyperthyroid patients. And so the reason that only 20 out of the 100 and some odd patients in the overall combined program on drug had sort of hyperthyroidism is because mostly the patients who came in were controlled. Now, many of them were controlled on moderate doses of ATDs and things like that, but nonetheless, like these were mostly not hyperthyroid patients at baseline. So, I think like from that perspective, they were less sick than the patients in the phase 2 study. I think we expect to show benefit in the Graves study on ocular symptoms, and we think it will be incrementally helpful. But, it's not obviously the focus of the Graves program, and the Graves program excludes moderate to severe TED patients who've already progressed to more significant ocular symptoms. Thank you for the questions.
Thank you. Thank you very much.
Thank you. And our next question comes from Chi Fong with Bank of America. Your line is open.
Hey guys, thanks for squeezing me in. I just have a quick follow-up on Brepo. So given the integrated trial design, and you mentioned parallel enrollment as well earlier in the call, how do you plan to handle the phase 2b results once you have those on hand? Could you or do you plan to top-line or publish the phase 2b results, or would the disclosure be more go or no-go or tweak or no tweak? And ultimately, the phase 2b data would be kept in-house until you have unblinded the phase 3 data. Thanks so much.
I don't know that we have a super well-formed view on what we'll say publicly about that data. I think our pretty strong expectation at some level, the biggest possible version of the impact of that data is like as some kind of futility analysis, basically, where like obviously if we saw something super unexpected, it could cause us to like change our conviction. I think that would be pretty surprising given what we know. And so, I think like that's sort of the sort of basics. Whether we will say something about it once we have it or not, to be totally honest, I just like haven't thought that much about it.
Okay, great. Thanks.
Thank you. I'm showing no further questions at this time. I'd like to turn the call over to Matt Gline for closing remarks.
Thanks very much. I appreciate it. Thank you, everybody, for dialing in. Obviously, a lot of good questions on both LPP and on Graves and TED. And sorry if we missed anyone in the queue. Look, I want to say thank you again. First of all, to the Immunovant team, to the patients and the investigators in the TED study. Obviously, always disappointing when a study doesn't work out. I hope one of the things you walk away from this call with is we learned a lot from that study that has been helpful. We learned even more that we haven't shared today because it's competitively valuable and will set us up to win in Graves. So excited for all of those learnings. And it's just a Herculean effort to get these things across the line. And then excited in advance from the same commitment that we're going to get from the LPP community, who are deeply in need of new therapies and really excited about the possibility of something new. I have great confidence in the Priovant team to run a good study there and to take advantage of all the relationships we've built and the work that we've done. And excited to just continue to add, big bricks in the wall that is the brepo opportunity. So looking forward to having a call similar to this one again in the not too distant future to talk more about new indications for brepo, at least on that side of it. Thanks, everybody. Thank you for joining this morning, and we'll talk again soon.
Thank you for your participation. You may now disconnect. Everyone, have a great day.